The aim of selective breeding is to introduce beneficial genes into an already desirable plant. An example of this is taking an apple variety that is good to eat but sensitive to disease, and breeding in the crabapple's disease resistance, but not its taste, size, and other traits. Traditionally, we do that by breeding or crossing the apple plant with a crabapple plant that has the disease-resistant gene we are interested in. Each offspring inherits genes from both parents, shown here as bars. Using DNA screening, we select the offspring with all the desired apple traits plus the crabapple disease resistance gene. This offspring is then crossed with the original apple parent. Once again, the offspring that has the disease resistance gene is selected and crossed again with the original apple parent. This is repeated over a number of generations, and with each generation the number of apple genes in the offspring increases, while the remaining crabapple genes decrease, until eventually we get a plant with most of its genes inherited from the apple plus the disease resistance gene. This whole process can take more than 20 years, as many plants require 4 years of growth between generations to produce the flowers necessary for breeding. With new breeding technologies, we can speed this up by modifying the flowering gene in the plant to make it flower in less time. The modified plant is crossed with a non-modified plant that has a gene we want. Let's call it gene A. In the first generation of offspring, some will have the modified flowering gene, some will have gene A, and some will have both. The offspring that have both are then crossed with a plant the same type as the modified parent, but it lacks the modified gene. From the second generation, we again select offspring with both the modified flowering gene and gene A. This is repeated through a number of generations, until the offspring mostly have genes inherited from the modified parent. From this generation, we select only offspring that have gene A but not the modified flowering gene for further development. These are called null segregants. By speeding up flowering through gene modification, we can produce a new or better variety in much less time.